Dear students, today's topic is enzymes, which previously completed polysaccharides and all. Enzymes. In this chapter, we are going to cover all enzyme and activity and all types. Enzymes are the biological catalytes, which influence the speed of biochemical reaction. So here, any one reaction has to be or the undergoes any changes required the catalyst, which are the enzymes. All enzymes are proteins, but all proteins are not enzymes, right? Here is the all enzymes are the, this line is very important. All enzymes are proteins, but all proteins are not enzymes, right? Enzymes are the specific, means each enzyme has a its own substrate. They will be work on the substrate, then they can be gives the product. That's the reason it is called as a, see for example, proteins, on the proteins proteases and lipids, lipase, right, like that will be there. Ribozymes, these are nucleic acids that behave like enzymes. They actually, ribozyme is a nucleic acid. It's behave like a enzyme, right? Nucleic acids, that's RNA, ribonucleic acid that behave like enzyme. This is a very, very important one. Ribozymes are the nucleic acids that behave like enzymes, right? Enzymes form tertiary structures, 3D structures with some crevices, means what pockets like structures called, we call as the active site into which substrate is fit. See here, for example, this is the enzyme. It has a, what is the structure of enzyme? Tertiary structure. Okay, in the 3D, here is the some crevices there, like a folds. Each fold has a, it has to be fixed the particular substrate. Then automatically that enzyme reaction will be takes. This is the substrate, this is the enzyme, lock and key, right? So here, what is the chemical reaction? Means what here is the on the substrate, for example, proteins. Okay, protease enzyme will be work on that. It will be changed into the amino acids, right? These chemical compounds undergoes are two types. Here is the physical change and chemical change. Two changes. The chemical change. Here is what the physical change. The change in shape or the state of matter without breaking bonds, right? Without breaking the particular matter, it will be change their shape. Best example, ice melts into water, water becomes vapor. Here what the thing, physical change means a change in a shape or a state of matter without breaking bonds. Ice will be changed into water without breaking bonds, and water also will be evaporate. These two are the physical changes. Chemical change means what? You know that these bonds are broken and new bonds are formed. For that, maybe organic or inorganic. Example, see, inorganic is the BaOH twice. Here is the H2SO4. When it's a BaSO4, bonds are breaking here, right? And sugar to glucose also, bronze bonds are breaking. But they have to memorize this one, organic and inorganic example, right? The rate of physical or chemical process is equal to amount of product formed. Okay, how can you determine this one? No, how much amount is produced, unit time, right? That is called as the physical or the chemical process, right? Now here are chemical reactions. Rate is called velocity. Okay, if the direction is specified, we know that velocity word. Rate of physical and chemical process are influenced by the factors such as the, what are the factors? Temperature will be essential. Rate of that doubles or the decreases by the half for every 10 degree centigrade. Every 10 degree centigrade. Got it? So here, the temperature, if it is low, the activity is a little bit slower. If the temperature is more, activity will be faster. We know that, right? So for that only, here what I'm going to explain here, that rate of physical or the chemical process are influenced by the temperature. If that rate doubles means or the decrease of means every 10 degrees centigrade, right? That's a one half rule you learned previously. Rate of enzyme activity, catalyzed reaction is higher in the carbonic anhydrase. You know that carbonic anhydrase Ca in the respiratory system, if it is present there, this will be 10 millions time is more. If it's not there, it's very slow. CO2 plus H2O in presence of carbonic anhydrase, H2CO3 will be formed. Okay, in absence of this enzyme, that's only 200 molecules are formed, right? Per hour, check it how it is. Difference is there. Per hour, it takes per 1 hour 200 molecules only will be formed H2O3. If the presence no, it will be as a 10 million times is a faster, right? In presence of that's a 6 lakh molecules are formed. See what the difference 200 molecules and 6 lakh molecules in absence and presence, right? That's the speed of reaction, right? See glucose and format two pyruvic acids in the glycolysis. And nature of enzyme action, catalytic enzyme cycle there, no, here is the enzyme, this is the E plus S, this is the enzyme and this is substrate, particular pockets that told no. In the pocket, this enzyme will be fits and form as a enzyme substrate complex. It will be after that it will be enzyme plus product will be formed, right? The pathway goes through the some unstable transition rate structures, right? EP means enzyme product complex is formed, right? And here is the concept of activation energy, how that energy is activated. How do enzymes speed up chemical reaction, right? For the purpose here, activation energy is like a starting initial energy. It is additional energy required for the start a reaction. If any chemical reaction want to start, required a particular energy that is called as the activation energy, right? In an exothermic and endothermic, two reactions are two kinds, exothermic and endothermic. In the it must be go through the much higher energy state. It is called as a transition state. One stage to other state, that a particular thing that will be formed, no, that is the transition state. Okay, activation energy is the difference between the average energy of substrate and the transition state energy. See here, if the product P at the lower energy, here they given as a substrate, here given S and transition state product. Here is the C from here to here is the substrate is very less, but its activation no, it reaches to the peak, right? That is the spontaneous reaction. It requires no energy by heating only happening. But biochemical reactions, enzymes lower the activation energy. As a result, speed of the reaction increases, right? That's called as the activation energy. I told you know here temperature and pH will be affect, activate, and concentration of substrate will be essential. Presence of inhibitor also will be activate there, right? What are the factors affecting the enzyme activity, right? These are the factors: temperature and pH, and concentration of the substrate, how much is there, and presence of inhibitor. These all will be effect on the factors on the enzyme activity. See here, temperature and pH. Enzyme shows the highest activity at the optimum temperature and pH, right? Meanwhile, here is the, see here, activation declines below the above optimal temperature, right? At high temperature, enzymes will be destroyed. This also, right? Because of the proteins are denatured by the heat. So temperature should be in a optimum way. Temperature should be in the optimum way. Or else no, if it is a more also have a issue and low also will be destroyed. Got it? And next one the here see inorganic catalysts work at high temperature, okay, and pressure. But the enzymes get denatured in the high temperature. If it's more than 40, no, they will be denatured. Thermophilous organisms, these are the exception, have enzymes with are stable at high temperature up to 100 also. Okay, Thermus aquaticus is these lives in the hot thermal springs, thermophilous bacteria, right? And here is the constant of substrate, concentration of substrate. The concentration of substrate increases, the enzyme that the pockets there, no, the sub enzyme will be fixed on that. With the increase in the substrate concentration, the velocity of the enzyme action rises at the first and reaches the maximum velocity, Vmax. We called as the Vmax. It will be reached the maximum. This is not exceed by the further rise concentration because of the enzyme molecules are fewer than the enzyme molecules, right? substrate molecule. Substrate molecule is a foreigner, then how where they should be fixed with that. That's why no free enzyme molecule to bind with the additional substrate molecule, right? Up to somehow this is the Vmax. After Vmax, there is no here they're gonna fewer than the substrate molecule. Substrate molecules are the fewer than the enzyme. Then where will the action takes place, right? And enzyme activity, presence of inhibitor. Inhibitor is a very essential. Enzyme inhibitor complex will be formed. Enzyme and substrate and inhibitor will come. Then what happens? Enzyme action will be destructed or the regulated by the these competitive inhibitors, right? The reaction has to goes on, but at a particular point it will be stopped because of these inhibitors, right? See, succinate, fumarate will be formed, succinic dehydrogenase. Okay, example of competitive inhibitor, best example, succinate. What is the inhibitor here? Succinic dehydrogenase. It form as a fumarate, right? And here is the different nomenclatures there in that chemistry what you learned, classification and nomenclature of enzymes, oxido reductases, dehydrogenases, and transferases, and hydrolyses, and liases, and isomerases, and ligases. These are they are given as a nomenclature, no substrate and reduced and oxidized and oxidized, and this is a reduced. Okay, these are the different classification of the nomenclature of enzymes. They have given as here translase and hydrolase. Okay, these are all are the nomenclature based on that. And cofactor. What is the cofactor? Here is these are non-protein constituents. These are non-protein bonds to the enzyme to make a enzyme catalytic activity. Apoenzyme is what? No, protein pot. Protein pot plus non-protein pot. Apoenzyme is the protein pot of the enzyme plus cofactor and hydrozyme. Here is the holoenzyme. Holoenzyme is there one pot. Holoenzyme means what? No, apoenzyme plus cofactor together called as the holoenzyme. Okay, cofactor plus apoenzyme is equal to holoenzyme. This is the inactive one and holoenzyme is the active one. When the cofactor is removed from the enzyme, this one if you removed, the catalytic activity is lost. That's why the cofactor has to be mixed with that and form as a holoenzyme, which is the active one. Right? Here is the objective is the apoenzyme is the protein part. Non-protein constituent as what? No, that's the cofactor, right? Here is the cofactors are the kinds. Prosthetic group is there, this is the one cofactor, and coenzyme is the one cofactor, and metal ions also cofactor, right? So here organic, these are the what are the prosthetic group? These are the organic form tightly bound to apoenzyme. Okay, the cofactor is tightly bound to the apoenzyme, and heme plus peroxidase catalyze. It is one of the organic prosthetic group, right? And coenzyme organic is the transient binding, okay, to the apoenzyme. Here is a what? Tightly binding. Here is a transient binding. Okay, coenzymes NAD and NADP, nicotinamide and dinucleotide and niacin are the coenzymes. And metal ions, these are the coordinate bonds with the side chains at the active sites and one or more coordination bond with the substrate. Example is the carboxypeptidase is the one of the metal ion. Zinc is the cofactor of this one, right? These things have to memorize because of only the asking, right? I hope you understood. Thank you. This all I make as a one playlist. If you want to, you have a interest to watch, you can watch and write a note in the under the heading biomolecule, that which shows the one short note like that. All files are there, all classes there. This is the biomolecule. If you are strong in chemistry, it's very easy. In Neet also, at least 4 questions they're asking every year. They should not be neglect. It's a key point to you. Thank you for watching.